Hey everybody, in this section I'm going to be going over cellular respiration. So probably just the very basis of cellular respiration is it's going to mostly be how our cells are going to get energy. You may remember ATP here is going to be the primary energy molecule for the human body. That's what all our cells are going to use. It's going to power our transport, it's going to power synthesis of pretty much everything. Super important molecule, ATP. So cellular respiration, what it is, it has this net equation, and it's going to take a C6H12O6, which is also, of course, glucose. And it's also going to take in oxygen, so O2, and then ADP, which is adenosine diphosphate, plus PI, which anytime you see PI in biology, that's going to be an inorganic phosphate. And from that one glucose molecule, 6 oxygen, ADP, and inorganic phosphate, there's going to be a very large amount of these. It can vary, though, depending on a couple things like the electron transport chain mainly. So you could get more ATP one cycle, maybe you'll get less the other. It does vary. Generally, I believe it's in the high 30s, though. But basically, you're going to take those reactants, and it's going to make carbon dioxide, 6 of that, which is what we exhale. It's also going to make 6 molecules of good old H2O, and then of course it's going to make ATP, which is what we are going to use as our primary energy source in the body. So cellular respiration is going to be divided up into a couple steps. The first of those steps, let me just get things centered here, is going to be glycolysis. So glycolysis, right off the bat, it's important to know glycolysis is happening in the cytosol of the cell, right? So this is going to be in the cytoplasm. It can occur really anywhere in the cytoplasm as long as your reactants are there, and you'll need the proper enzymes, of course. So sort of the net equation for glycolysis is going to be taking that glucose molecule, so that's C6H12O6, we're going to consume 2 ATP. We're going to take 2 molecules of NAD+, and from that, we're going to break that one glucose molecule into 2 C3H3O3s, which is also called pyruvate. It's going to make 4 ATP, and we're also going to get 2 NADH out of this, right? So from this equation, there are a couple important things. First, you can see that we both have ATP going in, and then a little more ATP coming out in the reactant side, right? So this 2 ATP is going to be in what's called the investment phase, and we'll demonstrate that down here. And then this 4 ATP is going to be our payoff phase. Other important things, you're going to see this NAD plus is going to turn into NADH. So we have this plus here, which means it has a positive 1 charge, and that's going away. Instead of that plus, we end up with an H. Now, the only way that that could happen is if we also gained an electron, because if you combine 1 proton and 1 electron, well, that's just hydrogen, right? Because a hydrogen has its 1 electron, and then, of course, it's 1 proton in the nucleus. So that is going to be a reduction. Super important to be able to track what is reduced and what is oxidized in a substance. Remember your oil rig, oxidation is loss of electrons and reduction is a gain of electrons. You can also think of it as reduction is reducing the charge. So we have a +1 charge and it gets reduced to a 0 charge. Reduction is usually pretty easy to spot. A little more difficult to spot is going to be this glucose molecule is actually going to be oxidized into these pyruvate molecules. Now, you can see if we look at this glucose molecule, we have a ratio of 1 carbon to 2 hydrogen to 1 oxygen. And then over here in the pyruvate, it's 1 carbon to 1 hydrogen to 1 oxygen, right? So we have lost hydrogen we've lost a little bit of everything because we split it in half, but we have lost hydrogen relative to the other elements, relative to the carbon and the oxygen. Typically, when you lose hydrogen in that ratio, it's going to be an oxidation. You can kind of think of it as that hydrogen, those hydrogens leaving, they're going to take their electrons with them. We're going to lose the electrons. So that's an oxidation. So that's pretty much everything from the equation. We have our inputs and our outputs. It's really important to try to remember inputs and outputs for each of the steps. That'd be a great thing to put down on your note sheets for the individual exam. So now, zoom in a little. We are going to go into the actual process. So I'm going over this process sort of for how we want you to know it for this class. There's going to be a ton of intermediates, a ton of enzymes that I skip. This is sort of just the big ideas. So first we're going to have this glucose molecule, and it is going to enter the cytoplasm. If you remember from the membrane transport chapter, glucose is going to enter through facilitated diffusion through a GLUT receptor. So we have this glucose molecule, and what's going to happen is a bunch of enzymes are going to act on it, rearrange it, but one super important change that is going to happen to this glucose molecule, which is a 6-carbon molecule, right? 1, 2, 3, 4, 5, and 6. We'll draw it like that. Those are each carbons. Is that's going to get converted into fructose, 1,6-bisphosphate. Bisphosphate just means that the phosphates are going to be on opposite ends. So now I have this 6-carbon fructose, which is an isomer to glucose, if you remember that. Not important to know that. But we are going to attach some phosphate groups. Ooh, I'm kind of squishing this in here. But we're going to have phosphates attached to each side. Now, where do we get those phosphates? That's going to come from ATP. So we're going to have 2 ATP, and there's going to be an enzyme that's going to cleave that 3rd phosphate from adenosine triphosphate to make them into adenosine diphosphates. This is going to be that investment, right? You got to spend money to make money is something that a lot of people say to help remember that. So we now have these 2 free phosphates that we got from this ATP, and that's what's going to go on to our fructose in these 2 phosphates, right? So now we've put our money in, we've spent the 2 ATP, right? So now we want to get that payoff. So now we're going to take 4 ADP, and it's also kind of important to note, a lot of this stuff is kind of just floating around in the cytoplasm. ADP and ATP, as we move through the term and the year, if you keep taking the track, ATP gets used for everything. So there's all kinds of ATP and ADP floating around, right? And there's even some AMP that'll become important in cell communication when we get to that. But we're going to take this 4 ATP and we're going to make it into 4 ATPs. This is going to be that payoff. So just remember, there's an investment phase where you lose 2 ATP, but then there's a payoff phase where we're gaining 4 ATP. And then in this step, there's also going to be 2 NAD pluses, and we're going to throw an electron on there. We'll throw 1 electron on each of those NAD pluses, and that's going to change those into these NADHs, right? So this process where we're turning fructose and ADP and ATP and NAD plus into NADH, this is going to result in this fructose 1,6-bisphosphate being split into 2 pyruvate molecules. So, and that's sort of the end product of glycolysis, right? We have these 2 pyruvates, which are these up here in the corner, C3H3O3s. And this has all been happening in the cytoplasm. So now we have these 2 pyruvate molecules, and now our cell kind of has a choice to make. It can either go the aerobic route, which means we are in the presence of oxygen, or it can take this pyruvate in the anaerobic route, which is when we do not have oxygen. So we're going to start with anaerobic because this is also going to be occurring in the cytoplasm, right? Zoom out just a little bit. Awesome. So if we go this anaerobic route, we are going to take this 2-pyruvate and we are just going to convert that into lactic acid, which you've probably heard lactic acid before. Lactic acid is that stuff that builds up if you're running sprints or you're lifting heavy weight. This is going to be what builds up because your muscles are going to use up their oxygen, and then it's going to switch to anaerobic and it's going to make lactic acid. There is a pretty common misunderstanding that lactic acid is what makes your muscles sore the next day. That is not the case. Lactic acid is an energy source, just like ATP is. Well, or rather just like pyruvate is. So in the process of converting this pyruvate to this lactic acid, for each pyruvate, and remember we have 2 pyruvate per glucose molecule, we are going to take 2 NADH and convert it back to NAD+. And you may remember from right over here, that's just the reverse of what we did to convert fructose 1,6-bisphosphate into pyruvate. So this process of NADH to NAD+ is referred to as NAD+ regeneration. That's pretty much all we need to know right now about the anaerobic pathway. Just remember, it happens in the cytosol and in the presence of no oxygen. But let's say that we are breathing super well and we are going to take the aerobic route. So if we're taking the aerobic route, we are going to head into the mitochondria. So, and remember, the mitochondria has this right here. This is the outer membrane, this sort of straight line. And then this wavy one is the inner membrane. And these are the cristae. Those sort of folds or indents are cristae. So we have outer membrane, intermembrane space, and then we are going to send this pyruvate to the mitochondrial matrix, which is sort of the lumen of the mitochondria. So then once we get that pyruvate in there through some transport, we are going to do what is called the transition phase or pyruvate oxidation. So this is a separate step from glycolysis. It kind of floats in this middle space between glycolysis and the next step, the Krebs cycle. Some people include it with glycolysis, some people include it with Krebs cycle. It's just important that you know that it is sort of its own distinct phase that is only going to be happening in aerobic conditions. So we are going to take this pyruvate, these 2 pyruvate that we have in the mitochondrial matrix, and we are going to add to them these coenzyme A's. So we have 2 coenzyme A's. Now, when we combine these 2 pyruvates and these 2 CoAs, we are going to get out of that. There's going to be, again, a bunch of enzymes that you don't need to worry about in this class that are going to spit out 2 carbon dioxide. And remember, everything that I'm saying is going to be per glucose. So for every 1 glucose, right here, we're going to get 2 carbon dioxide. All right, and then the other product and reactant that we're going to get is going to be this NAD+ is going to be reduced to NADH. Remember, it's reduction because we're losing this charge. So we're going to reduce 2 NAD+ to NADH. And the end product of this is going to be 2 molecules of acetyl coenzyme A or acetyl-CoA. And this is going to be the input of the Krebs cycle. So it's important to remember the end of glycolysis is pyruvate. And then if we go the aerobic route in the pyruvate oxidation or transition phase, we're going to end up with acetyl-CoA, which is going to be the input for the Krebs cycle. And just to sort of visualize the equation that we would have over here for the transition phase, we have 2 C3H3O3, which is our pyruvate, plus 2 coenzyme A's, plus 2 NADH, or NAD pluses, rather. So those 2 pyruvates are going to be oxidized into acetyl-CoA, and these 2 NAD pluses are going to be reduced to that NADH. So you may notice that we're getting a lot of NADH so far in this aerobic pathway, and that'll become really important later when we get to the electron transport chain, and we'll get to that later. But also, please note, I posted a video in the announcements maybe a week ago going over redox reactions, or it wasn't a video, it was a note sheet. Remember that oxidation and reduction occur simultaneously. So if something is reduced, then something else in the equation has to be oxidized. So in this case, pyruvate oxidized, NAD plus reduced. So that's going to be it for glycolysis. So now we are going to move on to the Krebs cycle, or TCA cycle. All right, so here we have the Krebs cycle, or the TCA cycle. It's important to remember this is also going to be happening in that mitochondrial matrix. So this is where the pyruvate oxidation happened as well. And when the Krebs cycle finishes, we are going to have a fully hydrolyzed glucose molecule. That means that glucose molecule that we put in at the beginning of glycolysis is going to be as broken down as it gets. So the Krebs cycle is a little bit hard conceptually because it's hard to understand, and there's just so many different enzymes. I tried to simplify it. I cut out, maybe 6 steps, which is 6 enzymes and 6 molecule names. But it's important just kind of know the main outcomes. So the Krebs cycle is going to do a lot of producing of NADH and FADH2. You may remember NADH is what we get when we reduce the NAD+, like we were doing in glycolysis and pyruvate oxidation. FADH2 is going to be a similar process because we're going to take FAD and reduce it to FADH2. Now, the reason that we are making that we want so much NADH and so much FADH2 is because when we get to the electron transport chain, we are going to need a lot of what is called reducing power. So when we think about that, reducing power means it has the ability to reduce well. So if we think about NADH, right? NADH has an electron that it kind of wants to give away because we stuck it with an electron when we took NAD+, and that made it NADH. So NADH and FADH are really good at giving away an electron, which is essentially what's going to power the next step of cellular respiration. They can donate electrons very well. So other sort of the big outcomes is going to be, this is where the most CO2 is going to get released as a waste product. And we are also going to generate a small amount of ATP here. And when I say a small amount, I am talking relative to the last step in cellular respiration, which is electron transport chain and chemiosmosis. But we are going to get a little bit. So sort of the equation is we're going to take that 2-acetyl-CoA that we had from pyruvate oxidation. We're going to take 6 NAD plus and 2 FAD, as well as 2 ADP. And you don't, by the way, need to memorize necessarily the numbers. It's more the concept of knowing that we're putting this NAD and this FAD to be reduced into NADH and FADH2. That's the goal. We just want you to know that this is a reduction, FAD to FADH2 and NAD plus to NADH, because that's the really important thing going forward into the electron transport cycle. Sorry, I cut out there. So remember, NAD plus reduced FAD is going to be reduced to FADH. ADP is going to become to ATP through phosphorylation. Phosphorylation is a word that you should get familiar with. All it means is the adding of a phosphate. And then this 2-acetyl-CoA that we had is going to be converted back into coenzyme A, and it's also going to make 4 molecules of carbon dioxide. And again, this is all per 1 glucose. So all I want you to know about the Krebs cycle is that we're going to take this acetyl-CoA and we are going to put it through this cycle that has all kinds of intermediates. It has, I could go through them, but it's honestly just confusing. But there are, what is it? I think there's 8 steps, and I included the first and last, which is citrate, and OAA, which is oxaloacetate. You don't need to know those necessarily, but this is a cycle. So it's important to remember that as long as we are getting the proper inputs, this can just keep turning. It can turn indefinitely because the end product gets converted into the start product. The only reason I included these two is because it's important to know that the Krebs cycle is going to slow down in the presence of high ATP. So the enzyme that's going to facilitate this oxalic acetate into citrate is inhibited when there's high levels of ATP. And this is called product inhibition. It means when the end result of this whole pathway, which is cellular respiration, because the end result was ATP, and that end result is going to inhibit further Krebs cycles from happening, which is good when you think about it, because if we have a bunch of ATP in our cell ready to go, we don't need to spend time, energy, or resources on making more until it's used up. So that's super important to know. And then just know as this cycle turns, it's going to spit out the 4 CO2, the 2 FADH, the 2 ATP, and the 6 NADH. That's pretty much it for Krebs cycle. Just remember, mitochondrial matrix, it's going to create a lot of reducing power in NADH and FADH2, and it's going to make CO2 as well as 2 ATP. So now we're going to get into sort of the final step of cellular respiration, which is going to be the electron transport chain, which oftentimes is shorted to ETC, and chemiosmosis. So I sort of mentioned it with the Krebs cycle. The electron transport chain is going to utilize all of that NADH and FADH2 that we have been building up from previous steps. It's going to use that reducing power to sort of power the machinery of the electron transport chain. And it's going to use that power to create a proton gradient, or H+. H+ is just a proton, right? So we are going to create an H+ gradient, and that gradient is going to power the ATP synthase, which is the machinery, the enzyme that is going to make ATP. Super important, probably going to be a test question. She loves asking this question. The machinery of the electron transport chain is embedded within the inner membrane of the mitochondria. So again, we have this big circle here. Let me scoot it up, which is the outer membrane. And then we have this wavy inner membrane here of the mitochondria with the cristae. And these pieces of machinery that make up the electron transport chain are actually embedded, they're transmembrane proteins on the inner mitochondrial membrane. Remember that. So I'm going to go through conceptually. This is not something where we're super concerned with thinking about inputs and outputs. It's very conceptual. So, and it looks really confusing, so I'm going to try to break it down in a way that makes sense. Remember, all in the matrix, all in this mitochondrial matrix, we did the pyruvate oxidation and we did the Krebs cycle, and we have just been producing a ton of NADH and FADH2. And remember, I've been saying that the reason we've done that is so that they can donate their electrons to the electron transport chain. So I don't want you guys to focus on memorizing any of the names of these. Some of them are easy. I don't want you to have to try to think of them as anything more than parts in a chain. We just want you to remember how the electrons sort of flow through it. So we have complex 1 and complex 2 here. And these are both going to take, so complex 2 is going to take the FADH, and complex 1 is going to take the NADH, and it's going to take electrons from them. So in complex 1, the NADH is going to convert back to NAD+. It's going to be oxidized to NAD+, and it's going to lose its electrons. So it's going to donate these electrons to complex 1. And FADH2 is going to do the same thing for complex 2, right? So now complex 1, complex 2 have these electrons in them, and these electrons are charged. They have this negative charge, and sort of the flow of electrons, as you might know, is electricity, right? And these properties are going to sort of make what's called a conformational change in the structure of these proteins. Specifically in complex 1 is what we're looking at. And what that conformational change is going to do is it's going to take protons from within the mitochondrial matrix, and it's going to pump them out of the mitochondrial matrix into the intermembrane space, so the space between the two membranes of the mitochondria. So we're using the reducing power of NADH, which means we're using its electrons, to pump these these protons into the intermembrane space, creating a high concentration of protons in the intermembrane space. And then all I really want you to think about is the electron transport chain is just kind of going to be playing hot potato with these electrons. Complex 1 and complex 2 are going to pass them to coenzyme Q, which is going to pass those electrons down the chain to complex 3. And here you see we have more hydrogen plus, so more protons being pumped into the intermembrane space, again, increasing that concentration. So now that we have this super high concentration of protons, it's just going to keep passing these electrons to cytochrome C, to complex 4, and that's going to pump some more protons out. So by the time we pass all those electrons to complex 4, we have a ton of protons in the intermembrane space. Tons. Big gradient. And we don't have many in the matrix because we've been pumping them out of the matrix. So the last thing that these electrons are going to do is they are going to be accepted by the final electron acceptor. And the final electron acceptor is always oxygen. Oxygen wants those electrons, and when oxygen picks up those electrons to sort of balance that charge, this oxygen is also going to pick up a few remaining protons in the matrix. And what happens when we combine electrons and protons? Well, remember, if we have one electron and we have one proton, and these cancel each other out, we're left with just hydrogen. And if we have hydrogen and oxygen being combined, that's going to become H2O, water. So this is where the water comes from in cellular respiration. So water comes from the electron transport chain. We're going to get 6 molecules of water per 1 glucose. So that is going to be it for the electron transport chain. Next, we're going to move on to the chemiosmosis. Remember, the end goal of the electron transport chain is that we have a high concentration of protons in the intermembrane space, and we have produced 6 water molecules. And now we are going to finish up the story of cellular respiration with chemiosmosis. So chemiosmosis is going to be where most of the ATP is going to be created in cellular respiration. Typically, you'll get 26 to 28 ATP per 1 glucose out of this step. All this step really is going to be ATP synthase, harnessing that very large proton gradient that we made. Because if you remember, we have a very high concentration from the electron transport chain. So now, just to orient you here, this here is the outer membrane, right? And then I actually drew out the phospholipids here of the inner membrane of the mitochondria. And then in here, we have the mitochondrial matrix. So embedded in the inner membrane, you have a bunch of these ATP synthases, which is what is going to make ATP. So it has a couple different subunits. It's a really, really cool molecule, and you can see really nice videos of it sort of doing its thing. It acts just like a rotary motor. So you're going to actually be able to see this bottom portion actually rotating around and sort of squishing ADP and inorganic phosphates into ATP. But as you remember, ATP has a lot of energy in that third bond between the second and third phosphate. So to make that ATP, we have to use energy. Because if we weren't using energy, that would just break the laws of thermodynamics, and that's a whole mess that we don't want to get into. So how do we get energy to make this happen? How do we get this thing to turn? Well, we use the proton gradient that we created in the electron transport chain. So all of these hydrogen pluses, which are protons, right? So all of these protons that are in the intermembrane space are going to flow back into the matrix through ATP synthase. Because remember, we have a very high concentration here and a very low concentration here, because we pumped, not only did we pump protons out of the matrix during ETC, we also used hydrogen pluses. We also used these protons to make water. So as these protons flow from a high concentration to a low concentration, they are actually going to generate a little bit of energy. And you kind of remember this if you watched the video that I did on secondary active transport when we talked about that. It's the same idea. ATP synthase is kind of like that secondary active transport in that it's using this gradient that we have established to power itself, right? So as these flow through, that tiny bit of energy that each one creates is going to be used to spin this bottom rotary part. And when that bottom rotary part spins, it's complicated to like visualize without the video, but basically it's going to squish an ADP and an inorganic phosphate. It's just going to push them together really hard into an ATP. And you can kind of think of it like you have your ATP, and between that second and third phosphate, I like to think of it as a spring. So this is the second phosphate, and then this is that third phosphate that we're adding to ADP. And you sort of have to compress them together so that they'll hold, but then we have a very tightly wound spring. So obviously when we release that last phosphate, that spring is going to really be able to generate a lot of energy. But first we have to compress it. And that compressing requires energy, and that energy comes from the proton gradient. So that is pretty much it for cellular respiration. Again, it's important to focus on inputs and outputs. We need you to know about redox, and then just sort of the goals of each step of cellular respiration. And then just for anyone who is sort of a equation-oriented person, for the electron transport chain and chemiosmosis, you do not have to memorize this one, but maybe it helps visualize it. We have 10 NADH, 2 FADH2, 6 oxygen, so 6 O2, plus we have our ADP and phosphates. And that NADH is going to get oxidized this time into NAD+. Remember, because oxidation is the loss of electrons, and we used those electrons to power the electron transport chain. Same thing with FAD, it's going to get oxidized too. And then we have O2 going to H2O, which is going to be our reduction, because we're gaining some electrons. Remember, we took those electrons and added them to oxygen, because oxygen is the final electron acceptor. Can't say it enough, probably will be a test question. So that is pretty much it for cellular respiration. I hope you guys found this one helpful, and I will hopefully see you in the next section.